This laboratory will take place over the course of the next 2 weeks. The Monday session of each week will consist of inoculating various mediums, with each Wednesday session being a results day. The cultures you will be using will be located on your bench tops. It is extremely important that you label your tubes properly. Most of the mediums we will cover during the next 2 weeks will be used to determine the identities of your 2 microorganisms for your unknown project. Care should be taken in learning the reason for doing a test, as well as how to read the result. Let's now briefly discuss what you will be working with for day 1. Just like we need carbohydrates to provide us with energy, bacteria also utilize carbohydrates for the same reason. The carbohydrate test is usually a good place to start when examining how an organism metabolizes glucose, sucrose, or lactose. Glucose is a simple sugar, a monosaccharide, while sucrose and lactose are complex sugars, or disaccharides. Furthermore, for an organism to break down lactose, it needs to possess the enzyme beta-galactosidase. Carbohydrate tubes have 2 important qualities. First, they possess a pH indicator, phenol red, which turns yellow at acidic pH, and second, they contain an inverted Durham tube. The Durham tube allows for any gas production to be collected in the carbohydrate tube. Therefore, an organism that possesses the enzyme to break down a carbohydrate may produce acid or acid and gas as byproducts of the organism's fermentation of the sugar. On the other hand, if an organism is unable to break down a carbohydrate, it lacks the enzyme necessary and will cause no change in the culture medium, and the medium will be red with no gas bubble present in the Durham tube. The next test is the starch test. Organisms that possess the ability to produce extracellular enzymes will test positive on this medium. Starch is a complex carbohydrate made up of repeating linked glucose molecules. In this exercise, if an organism is able to hydrolyze the starch by producing an extracellular enzyme, we will be able to detect this by subjecting the plate to iodine crystals. When inoculating this plate with an inoculating loop, it is recommended to simply draw a line in the middle of the plate. That way, you have an area of inoculation as well as an area where no organism has been placed. After growth has occurred, a plate of starch agar is inverted over iodine crystals. After 1 minute or so of exposure, you can view the result. Organisms that are able to produce alpha amylase secrete the enzyme in the surrounding medium, which will result in a clear zone around the area of growth. An organism that is unable to make this enzyme will have no clear zone, and the purple color that is produced is from the reaction of the iodine vapor and the starch still present in the plate. Some organisms are able to produce extracellular enzymes that break down proteins, which leads us to the next 2 tests. Milk contains a protein that certain microorganisms can break down. This protein is casein and is the reason milk has its white appearance. These organisms produce extracellular proteolytic enzymes, which reduces these proteins to their individual subunits, which we know are amino acids. When inoculating this plate, it is recommended to simply draw a line in the middle of the plate with an inoculating loop. That way, you have an area with an organism as well as an area where no organism has been placed. After this medium has been incubated for 2 days, you can view the result. Organisms that have the ability to break down casein will appear to have a clearing of the medium around the area of inoculation, and those that do not will appear as if no change to the medium has occurred. Another protein is gelatin, which is added to a medium as a solidifying agent. Gelatin is derived from the animal protein collagen. This protein is the component of connective tissues and tendons. Gelatin is liquefied in the incubator, but can solidify when cooled below 25 degrees Celsius. The gelatin hydrolysis test is used to determine if an organism possesses the ability to produce gelatinase, which is excreted from the bacterial cell. This test is conducted by using stab inoculation. After stabbing this medium with the organism, the medium can take up to 1 week to see a result. Therefore, it is not recommended to use this test unless you have the time to do so. After incubation, care must be taken to not shake the tube, as you will mix the hydrolyzed with the underhydrolyzed gelatin, which will give you a false negative result. To develop this test, you will simply place the tube in the refrigerator for 5 or so minutes until the control tube becomes solid. Once the control tube is solid, remove your tube from the rack and carefully tip your tube to the side, being careful not to dump the contents of your tube. If the medium does not tip when this is done, it is a negative result. If the contents of the tube tips with the tube, then the organism tested produces proteolytic enzymes. The last medium we'll be using today is the blood agar plate. These plates are made with TSA, not the Transportation Safety Administration. They had no hand in this, but rather tryptocase soy agar, which contains 5% sheep's blood. This medium is found in most clinical microbiology laboratories, as it is used to test for an organism's ability to produce hemolysis. Hemolysis can break down, as the name suggests, blood by lysing them. There are 3 types of hemolytic reactions, these being alpha, beta, and gamma. Alpha hemolysis is a result of incomplete or partial breakdown of the red blood cells and appears as a green color. Beta hemolysis is a result of complete breakdown of the red blood cells, resulting in a clearing in the medium. And lastly, gamma hemolysis is no breakdown of red blood cells and often appears as a browning of the medium. There will be no noticeable change in the medium if the organism does not produce hemolysis. Therefore, any organism that possesses the ability to break down red blood cells can be problematic. Today, you and your partner will be conducting a nasal and throat swab on 2 separate plates. Divide your plates in half and you will streak using a cotton tip applicator your nose on one side of the plate. To open the swab, grab the side that has no cotton and simply pull down on the paper wrapping and remove the applicator. You can obtain your nasal sample yourself and should place the swab into your nose until you feel slight discomfort, but nothing more. We don't want you to damage those precious brains. To conduct your throat swab, you will need to employ the services of your lab partner. You will say, "ah," and they will swab the back area of your throat, specifically the tonsillar area, which is back where your uvula is, but on the sides of your throat. Care should be taken not to touch the tongue or the roof of the mouth. Both swabs should be treated as biohazard waste and disposed of in the biohazard trash. You will not reuse your swabs. Next lab, I hope you enjoy seeing the organisms that you harbor in your own bodies.